Hey guys, we're back and we're talking about the central dogma today. So last time we talked about the two types of nucleic acids, which is what we're talking about here in this first box, just a review of how they're different. And remember, we have DNA and we have RNA. And I'm going to carry those colors that I used last time throughout this sheet as well. So purple for DNA and orange for RNA. And today we're going to talk about how the DNA and RNA work together to actually build our proteins. And remember, proteins are another macromolecule that we've talked about this year, and proteins do all your cells' jobs. So they're going to be enzymes to speed up reactions. They're going to be structural, like your muscles. They're going to be in your organs and your heart and all of the things. So proteins do a ton of different jobs. They're in your cell membrane. They let things in and out that are too big for the phospholipids. So proteins are super duper important. And remember that DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose sugar, that's our D, and RNA has ribose sugar, that's our R. Now this is the one that's going to be important for today, is that DNA uses thymine and RNA uses uracil. We have to remember that because when we get to protein synthesis and we're looking at how to read DNA to give us RNA, you're going to have to remember that that base pairing rule is going to switch a little bit depending on if you're going DNA to DNA or DNA to RNA. And then DNA lives in the nucleus, and we're talking about eukaryotic cells here because eukaryotic cells have a nucleus. Prokaryotes like bacteria are going to do this a little bit different. So remember that we're talking about eukaryotic cells. And then RNA can move between the nucleus and the cell because it's the messenger. How do we read RNA? RNA is a nucleic acid, and RNA is a code for a single protein. So if you're going to make a note, please make a note that one RNA strand equals one protein. Remember our cookbook versus recipe analogy? Remember, DNA is the cookbook, so it has all of the pages, all of the recipes, all of the instructions. RNA is the message telling us how to cook one recipe or how to build one protein. And then we read RNA in 3-letter chunks called a codon. So 3 letters, codes for an amino acid. And remember, amino acids are the building blocks of proteins. So we read RNA in codons. So if you look here, we would have this strand of RNA with all the bases. And remember, we've used U instead of T. And each 3-letter set is a codon that will give us one piece of the protein we're building. When we talk about the central dogma, this picture is the explanation of start to finish. We have DNA replication, which is DNA to DNA when we make a copy of the whole DNA strand. We have transcription, which is DNA to RNA, which we are going to focus on today, where DNA goes and we transfer all of those bases into the RNA code. Remember, we use U instead of T. You can also do reverse transcription, where you take an RNA and figure out what the DNA was by going backwards. Then we have translation. And I always think about translation versus transcription, as transcription is the same language. We're still in nucleic acid. DNA to RNA is nucleic acid to nucleic acid. So we're just rewriting it. Translation is a nucleic acid to a protein. So we're going from RNA to amino acids, which are the building blocks of proteins. So we're actually changing languages. We're changing macromolecules. So transcription is rewriting. Translation is a new language. And then once we have this amino acid strand built from the RNA, we then have to do a little bit more work to the protein to make it actually function. And we have to fold and spiral and pack in so that the protein can do its job. So this is the central dogma, DNA to protein. The first step of going from DNA to protein is actually building our RNA molecule, and we do that in transcription. So transcription is the making of RNA from DNA. This is going to happen in the nucleus because DNA can't leave the nucleus. DNA has to be done, any of the work it's going to do inside the nucleus. So when we're in the nucleus, DNA is actually going to unzip. We're going to read the section of DNA that we want to build the protein from, and that's a gene. We're going to make a copy of those letters, and when we make that copy, we are making RNA. But when we make the copy, remember that A is going to go to U. So when we see an A in DNA, we write a U for RNA. Now there's going to be T's in our DNA, and T still gives us an A because A is universal between the two. It's when we see an A, we write a U. Now, how does this happen? First, we have to split the DNA because remember, DNA is double-stranded and it is held together by those weak hydrogen bonds. And when DNA is zipped up or held together, you can't read it. So the first step is to break those hydrogen bonds. Notice we have this little pocket here. So this is our gene that we're trying to copy. And we've popped that DNA open, kind of like a Ziploc baggie. You pop it open. It's still held together at the ends, but the middle is open. That is our gene that we're going to replicate or transcribe. And that's where we make our RNA. So we put those complementary RNA nucleotides onto the DNA so that we can make our copy here. Notice A goes to U. And then that RNA will detach. It will leave the nucleus and the DNA will zip back up and those bonds will come back together. So just to practice that idea, we have a DNA strand. And how do you transcribe? You write the complementary pairs. So if you see an A, you write a U. You see a T, you write an A. T, A. C, G, G, C, and so on. Now this is where we get to that reverse transcription. If for some reason you have the RNA strand, but you want to know what the DNA gene was, you can go backwards. So if you see a U, you would write an A. A, you would write a T. Notice that here I wrote T and here I wrote U. That's because I'm paying attention. I'm writing RNA in transcription, so I use U. I'm writing DNA in reverse transcription, so I'm writing T. So depending on what I've been asked for is the letter that I use. So when you're looking at questions and practices for this section, make sure you're really careful about what you're writing, if it's DNA or if it's RNA. Next, we're going to go to transcription. And we're going to go to translation. We just finished transcription. Translation is where we go from RNA to protein. So we're changing languages. We're changing macromolecules. So the process of translation is to read RNA and to use that code or the codons, the three-letter chunks, to build a chain of amino acids, which is our protein. This is going to happen in the cytoplasm, so that jelly of the cell, and it's done by a ribosome. Remember, ribosomes build proteins. So we have two macromolecules, nucleic acids and proteins, and we have two organelles, the cytoplasm and the ribosome. Once mRNA is made in the nucleus and sent out into the cell, a ribosome will attach to that message because it's mRNA, the messenger RNA. The ribosome is going to read that message in 3-letter pieces called codons, and each 3-letter piece is going to tell us which amino acid will be transferred or put onto the chain. And we read RNA using anticodons, so the complementary to the letters on the message. And you can see that in this picture. So we have our transfer RNA that have the anticodon that is complementary to the codon on the messenger RNA. As these transfer RNAs come in, they are going to build this chain of amino acids, and each code tells us a specific amino acid, and those amino acids are held together by peptide bonds. So proteins are held together by peptide bonds, P to P, protein peptide. And peptide bonds are strong bonds. They're not going to be easily broken. And once we have built this chain, we can then express the gene or do whatever the recipe was telling us. That's the goal of the central dogma, is to go from DNA all the way to the functioning protein. Now, here's where it gets tricky. How do you actually read those codons? So we're going to do this one together, and I know you guys like to just write the notes, but please take the time, pause, follow what I'm doing, use the codon chart here, and get used to how to read that, because you are going to be asked to do that on your practices and your test. So I've done the first one here, and I've kind of shown you how this circle works. So our first codon is AUG, and you're going to read the codons in the circle chart big to little. So our first letter is A, so I'm in this quadrant of the circle. My second letter is U, so then I go to this piece of the pie, and my third letter is G, so then I'm at this sliver or slice. So I go big, medium, little. This quadrant to this piece to this slice. Now, when you write the amino acid, which are these big fancy words that all end in INE, those are going to be your amino acids that are going to build the protein. There are 20 options for amino acids. And you'll notice that some of them are repeated on here, like leucine is on both sides. That's because you have this effect where 2 codons can code for the same amino acid, and that's just a fail-safe, and we'll talk about that more when we get to mutations. But you'll notice that it has the full word, methionine, and then it has this little abbreviation up here, MET. Those are what we want you to write when you're doing your process. I don't want you to write the whole big long word every time, but I do want you to write the 3-letter code, because this big letter on the outside doesn't really stick the same way as the 3 letters. You're going to know that met versus li versus lu is more beneficial to you than the big letters. So we're going to use the 3-letter code. So met is my first amino acid. Our second codon is GGC. So I'm going to go to the big G. I'm going to go to the medium G. And I'm going to go to the C. And that C, all 4 of our little letters for this one code for glycine. And my 3-letter abbreviation is GLY. My second codon is UAC. So I'm going to go to the big U, the medium A, and the little c. Notice that the little c goes to tyrosine. So my code here is TYR. Then I have CCU. Big C, medium C, little u. Codes for proline, my code is PRO. Then my last codon is UAG. UAG. Notice that it's stopped. That means it's the end of the protein. So you can either write stop or you can use the abbreviation STP. Doesn't matter. Now, this is an important thing to remember also. Met is our start. So sometimes you're going to see them write met, sometimes you're going to see them write start, but either way is okay. Stop is always going to be stop. Now go ahead and pause the video, do the other two examples, and come back and check your work off of mine in just a second. All right, you should be back with us now. You should be checking to make sure you got all the correct amino acids. If you messed up, that is perfectly fine. Just cross it out and fix it. We're going to be practicing this a lot. If you have any questions, please make sure you ask for your teacher to show you how to use this codon chart in person, because it is going to be a big part of your practice and test for this unit. And then we get to our final part of protein synthesis and the central dogma. And that's, how do we get a functioning protein? We start in the nucleus. The recipe is copied. This is going to be transcription. Notice that I kept that orange from transcription. Then we move into the cell and the ribosome attaches to the message and we go through translation. So we take RNA and turn it into the amino acid chain. So we've read the recipe and put all the ingredients together. Once the chain of amino acids is made, it's going to move to the rough ER. And remember, there are two types of ER, or endoplasmic reticulum. And the rough ER has ribosomes attached. So it is a transport system for proteins. And this is where we're going to move the food to the stove to cook it, where we're getting ready to make the dinner. The Golgi body is the final prep step. We're going to cook the meal. We're going to get everything perfect and ready to use. And then we ship the meal off to be eaten or taste tested. And that's going to happen in the cell membrane. So all proteins have to be proofread before they're let out into the cell to do their job, because if they're not right, they won't do their job properly. So the cell membrane checks to make sure that the protein is built correctly. If it is correct, it sends it off to wherever it needs to go to do its job. If it is incorrect, we jump down here to the lysosome. And remember, lysosome stands for Lysol. It's the cleaner. If it's incorrect and it's not going to work, the lysosome is going to break it down, put the parts back out there, and let it be rebuilt. So a protein has a lot of steps before it is ready to be used. And that makes sense, right? Because proteins are doing so many of your cells' jobs and your body's jobs that if they're not perfect, that's going to mess up how your cells in your body are functioning. So we go all the way from nucleus to cell membrane before that protein is allowed to do its job. All right, check your modules. Make sure you do your practice for today. And if you have any questions, please, please, please ask your teacher.